Hi, everyone. Welcome. My name is Rebecca Lee, and I'm really delighted to be here at the kickoff of our AMR Register. I'm the Executive Director of Vivli, and I'm really delighted to have all of you here today. Joining me here is Tim Jinks. Tim, would you like to unmute your camera? Tim will be giving opening remarks. Tim is Head of Infectious Diseases at the Wellcome Trust. Wellcome provided the seed money for our AMR Register, and we're very grateful for the support of Wellcome in helping us lift the AMR Register. So Tim, I turn it over to you.
Great, thank you, Rebecca. It's a real pleasure to be here, and I'm absolutely thrilled to see the launch of the AMR Register as part of the Vivli platform. There's an important issue as relates to AMR, and there's also the more general issue as relates to data sharing. And I want to touch on each of those briefly. AMR, as we've seen in the recently reported Burden of Disease project from the Graham study, in 2019, over 1.27 million people worldwide died directly as a result of AMR. And overall, 4.95 million people died with an AMR type infection, a drug-resistant infection. These results tell us actually the scale of the urgency worldwide. It's much worse than had been previously understood or modeled. The numbers that are off reported are those that were generated by the O'Neill review, the review on AMR. But now we can see with this revised modeling and estimates that the situation is very significant and increasing. So the urgency of AMR as a problem, as a global problem, is very real and one that demands attention and action worldwide. This project is something that can help to contribute toward that. And indeed, in fact, there was a lot of industry generated data that was donated to the researchers that did the studies and the modeling in that Graham report. And that demonstrates the utility, the applicability and utility of this type of shared data. Sharing data and reusing data, that was an important thing for actually creating a public good. It's important that there are these assets which are generated for other purposes, that they are managed through a long-term life cycle. And of course, that is done in a way that is ethical, transparent, and legally compliant. Really important, though, is that the data can be brought together in a way so that there are common data terms and standards, and promote the usefulness of being able to interrogate and use the data by researchers and others. And lastly, there are elements of security that have to be done to make sure that the data is accessed by appropriate governance and rules, and certainly not misused in any way or ways that were not authorized at the source of generation, because indeed a lot of AMR data that's quite informative ties back to patients overall, something that we hope to see. So really, we want to make sure that data reuse is something that can be done to inform evidence for action with respect to the AMR threat as a whole, but also this is something that is important in more broad terms, that data reuse is something that should be done wherever it is appropriate and feasible, so that we can have these sorts of public goods that are more effective utilization of resources that are generated worldwide. So really great to see that Vivli have arrived at the point of being able to launch the AMR Register in their platform. I'm certain that the provision of data in this way will be a useful tool for researchers and others who are wanting to understand what's happening in terms of transmission of antimicrobial resistance. And really important here that we recognize that this is a non-trivial undertaking overall. The technical aspects behind the scenes are a difficult one, and congratulations to the Vivly team for addressing that. Also, very, very important that the private sector players who have provided the data do so. This is something that, you know, it isn't required. I think that it is something that almost should be, but of course it has to be done on a basis that's appropriate, fair, and reasonable to private sector players who are doing this. But huge thanks go out to each of the companies who have donated their data to the AMR Register as well. So with that, I just want to thank you for allowing me to participate, and I look forward to seeing the launch.
Thanks so much, Tim. Really delighted to have you here today. What I'd like to do next is just step through the agenda and an introduction to the register itself. Thank you for your opening remarks. Today, I'd like to give you an introduction, a broad introduction to the register itself for a few minutes. Then we'll see a live demonstration of the register given by Stan Newman, our Director of Technical Operations, and Patricia Bradford, our microbiology consultant. We'll have an introduction to the data sets themselves by our data contributors, and then we'll close with some Q&A. As we go through, feel free to put your questions in the question and answer box. What I'd like to do next is really introduce our team that's been behind this for so long. So as I do so, I'd like our team to turn on their video. This has really been a team effort, and I'm very grateful for the team that has been behind this. So Nina Hill, our AMR consultant. Thank you, Nina. You've just been really helpful to help lead this effort. Patricia Bradford, our microbiology consultant. Catherine Darcy, who's been managing this program, and Stan Newman, our technical lead, who's been leading the team from a technical perspective and overseeing Insight, our architect, on this. So thank you, team. I'm really delighted to be part of this team that has been working so tirelessly over the past 18 months. Also, I'd like to introduce our scientific advisory board, who is led by Mark Mendelson at the University of Cape Town. Our SAB has really been advising us in quite a few of the scientific aspects, but primarily to assist with outreach to the scientific community and outreach to researchers. They will continue to do so, and also advising us on the value proposition. Many of them are here today. When we think about the value proposition of the AMR Register itself, until today, really, there's been no single unifying platform for high-quality industry surveillance data. There have been many other high-quality AMR registers to share data, but there's no single unifying platform until today, and that's really exciting to be here at the kickoff of this register. So this register will enable detecting trends in multi-drug resistance over time, informing national and international policy on antibiotic stewardship, and also allowing modeling of future resistance trends. How the AMR Register works, for those of you looking and seeking to access this data, is first to search the register for surveillance data sets, and you'll be seeing this live of how you can do this, making a request for data sets, accessing these data sets through downloading the data, analyzing it, and then going on to publish your results. So how it would work is you come in and do a search for those data sets of interest to you, accessing those data sets, and then going on to integrate the data and doing your analyses. So I just clicked ahead. Some benefits for those of you who might be looking to contribute data, and those of you that have already contributed data include advancing the science in antimicrobial stewardship, increasing discoverability by sharing that data through an AMR platform, and also meeting publicly stated commitments. Benefits for researchers, so those of you who are researchers who might be listening in, really include accessing this high quality industry data in a single centralized platform to aid in critical decision making. So the data in our platform today, and we certainly are looking to expand that, include over 925,000 isolates collected over the years 2004 and 2020 from over 80 countries, testing 69 antimicrobials on 434 organisms. These include from over 40 LMICs, and these are listed here. I'm really proud to introduce our founding steering committee members. These have worked really hard over the last 18 months to help us with governance, and many of them are here today to introduce the data sets that they've contributed, and also the why behind why they're working with us today. These companies have really been generating local antibiotic susceptibility data for a long time. They monitor pathogens and changes in resistance patterns through centralized testing using internationally accepted reference standards. In 2016, the AMR Industry Alliance members really committed to continuing to share the surveillance data that they generate with others. And I think this is really the fruition of some of that. And we really are proud to work with many of these founding steering committee members to move that forward today. So looking at what we'd like to show you next is a demonstration of this AMR register, a search for data sets that you will see in this unifying platform, how to request data sets, and then accessing data sets of interest via the AMR register. So Stan and Patricia will be doing this live demonstration of the register. I'll turn it over to them. So Stan and Patricia.
Good morning. So are you seeing the AMR register itself? Let me confirm. Okay. So as Rebecca suggested, we're going to take a look at the live platform from the perspective of the researcher who's looking for data. Now, their experience begins with the process of searching for data sets that can contribute to the research topic that they're about to embark on. The search screen, which is displayed here, gives the researcher a number of choices to find appropriate data sets. Let's start by clicking on Browse. This brings me to the Research Programs page. This page lists all of the data sets that are available through the AMR register so that the researcher can explore each program and decide whether it'll meet their research criteria. Back at the search screen, if I click on the magnifying glass over here on the right, or the number of data sets at the bottom, I'll be taken to the complete list of data sets that are available, and I can select the ones that I'm interested in. We'll come back in a moment to discuss how to determine which data sets I might be interested in. Alternatively, back at the search screen, I can choose to filter the list of data sets using the drop-down list here to refine the list for my particular research. At this point, I'll invite Patricia to describe a few research questions that the system can help to address.
Thank you, Stan. First, we're going to do a search for something that really everyone is interested in, and that's carbapenem resistance. So first, from antimicrobials, we're going to pick meropenem, and then organism Klebsiella pneumoniae. And during these searches, you can choose as many search terms as you like. Today, we're interested in the region of North America. And I'm interested in lower respiratory tract infections, so I'll pick bronchoalveolar lavage as for the specimen. And we see that there's 3 data sets that contain that information, and they're listed there. they have different attributes. The next search that we're going to do is just to show you that really how very specific you can get, and that there's data on some countries where you may we may not always see a lot of data. So organism, please pick E. coli, Escherichia coli. And then today I'm interested in the country of Qatar. You'll see from the country list as Stan is scrolling how many different countries are available, and that some of them are, really of interest hyperlocally. So we see that there's one country, there's one data set that has that requirement of information from Qatar. Thank you. So the next search, we want to look for MRSA. So we have search terms for some resistance groupings that have MRSA. So please pick that. And we'll see that there's only one data set that contains that resistance grouping. And I just want to mention here that this particular data set is the only one that came pre-specified with MRSA as a column in their data set. But if you're interested in MRSA, there's another way to do it too. So now we're going to go antimicrobials and look for oxacillin. And when you receive these data sets, you can use the MICs of oxacillin to choose the MRSA. And then Staph aureus, Staphylococcus aureus from organisms. And now we see that there's two data sets. So the first one we found before, the second one came up because it has oxacillin MICs and Staph aureus, and you could use that information then to select the MRSA from that data set. That's it. Thank you.
Okay, thank you. So at this point, if we want to explore further to determine whether any of these data sets will actually be useful to me, I can click on View Dataset Details. The system displays a page with additional information about this data set, including the dates it was collected, the number of isolates, the protocol summary, and a variety of other information about this data set. If the contributor of this data set has any additional documents that might be useful to a person who is considering this, they could place it here in the data set documents. Moving on to administrative details, we can see that the system has generated a digital object identifier. This is a unique identifier that can be used to reference this data set in any publications. And finally, we're tracking the usage. This is based on a broadly used standard called the Counter Code of Practice. At the moment, because this is a brand new system, nobody has had access to it, so our counts are all 0. But once the system has been in use for a while, we will see under the views this number of people who have viewed the metadata for this data set. If there is an additional document, we'll count the number of times those were downloaded. And in particular, probably of more interest, the number of times someone has actually accessed the data and downloaded it. Now, to give you some basis for understanding these numbers, we'll include the range of dates over which those have been collected. Now, at the moment, of course, we're looking at this from the standpoint of a researcher. Needless to say, this information is also available to the data contributors. If I decide that this data set is useful for my research, I can simply request it. System will give me a list of any research projects I've already started. In this case, I will add a new one. Looking for the distribution of MRSA, and the system will add it to a project. I can continue to look for and select data sets that might be part of my research. When I'm done, I can click on My Data Requests. System will show me that project that I had started to formulate, and I can click on that. And the system opens up a form that allows me to describe my research project. Now you notice a number of the fields are highlighted in red. These are required fields that have not had a value provided. As I type stuff in, you'll see the red disappear. So first of all, we need to know the researcher's name, email address, their affiliation. I can supply an ORC ID, specify my location. I'm in the United States of America, so I will select that. The system asks me to acknowledge that if I create any publications from this research, that I will acknowledge where the data came from. I need to agree to the terms of use of the data. I need to provide a summary of the research, and I will paste it so you don't have to watch me type. And then lastly, I need to identify the purposes of the analysis. Now at this point, the submit button's enabled. I can click submit. This makes the request available to the system after a very brief period of administrative processing, usually less than a day. I, as a researcher, will receive an email notification letting me know that the data is available. At that point, I'll reconnect to the system. I'll go to the Active tab, and the Fulfilled request is sitting here. If I open that, I'm first presented with a form which basically summarizes the information that I filled out when I was requesting this data set. More interestingly, if I go to the Datasets tab, I will see those data sets that I've requested. If I click on one of them, system will give me a form that actually looks somewhat familiar. This is essentially the form that I used when I was deciding whether this data set would be useful to me. The particularly noteworthy is that we now have a download tab. If I go to the download tab, I can see the data set itself. Simply click on the download button, and the system will download that to my browser, through my browser to my laptop. And I can simply click on the file and open it up, and I have the data that I have requested. And at this point, I can begin my research topic. At that point, I will hand things back to Rebecca for the next stage.
Thank you so much, Stan and Patricia. So what I'd like to do next is introduce those steering committee members that are here. As I read your company name, if you could turn on your video and introduce yourself. So I'll just go down the line here. Paratech.
Hi, good morning, good afternoon. I'm Elisa Sirio. I'm happy to be here, thanks.
Thanks, Elisa. Shinogi.
Hi there, good afternoon or good morning. I'm Chris Longshaw, Senior Director of Scientific Affairs for Shinogi.
Hi, Chris. Venatorix.
Yes, hello everyone. It's Greg Mowick from Venatorix, Senior VP of Microbiology. Very happy to be with you today.
Hi, Greg. Merck team.
Good morning, everyone. Michael Wong from Scientific Affairs with Merck.
Mary Model, Clinical Microbiologist.
Fakhar Siddiqui, Global Director of Medical Affairs.
Thank you, Fakhar. Pfizer.
Thanks, Rebecca. Hi, everyone. It's great to be here. I'm Gemma Buckland-Merritt, External Medical Engagement Lead from Pfizer.
Thank you, Gemma. J&J.
Hi, Rebecca. Carla Childers here, Bioethics and Strategic Projects Leader for Johnson & Johnson in the Office of the Chief Medical Officer. Thanks for having me.
Thank you, Carla. And GSK?
Hello, Lovely to be here. Thank you. It's Karen Langfeld from Global Medical Affairs at GSK.
Terrific. Thank you so much. So next, what we'll move to is the presentations by our data contributors. Some will be presenting videos, and some will be presenting short slide presentations. Please feel free to put questions and answers, questions in the question box, and then our panels will answer them at the end of this segment. Thank you so much.
Give me 1 second. Let me go.
So right now you'll see a short series of videos.
I'm Elisa Serio, Senior Director of Microbiology at Paratech. Antimicrobial resistance is an urgent and growing public health threat that continues to rise worldwide, leaving limited treatment options for patients with even the most common infections. Paratech is a biopharmaceutical company focused on the development and commercialization of novel life-saving therapies to combat life-threatening diseases as well as other public health threats for civilian, government, and military use. We are members of the Antimicrobials Working Group and are proud to join Vivli's new Antimicrobial Resistance Register. Our commercial product, omadacycline, which is marketed under the trade name Nuzyra, is a broad-spectrum, modernized tetracycline class agent specifically designed to overcome the most common tetracycline resistance mechanisms. Nuzyra is FDA approved for the treatment of adults with acute bacterial skin and skin structure infections and community-acquired bacterial pneumonia. Paratech has taken part in a robust annual surveillance program since 2009 called Keystone, which is part of the broader Sentry surveillance program conducted by JMI Laboratories. Keystone was designed to monitor omadacycline susceptibility over time while fulfilling post-marketing regulatory requirements. Our submission to the Vivli AMR Register will contain the in vitro activity of omadacycline and comparator agents against over 60,000 gram-positive and gram-negative isolates that were collected from diverse infection types during 2014 to 2020 throughout the United States and Europe. Additionally, we are excited to provide links to the Keystone Surveillance website and recent omadacycline surveillance publications. We are proud to be part of Vivli's efforts in advancing human health and antimicrobial stewardship through this new and exciting AMR data sharing platform. Thank you.
Hello, my name is Chris Longshaw from Shinogi, and I'd like to say a few words about why we are very excited to be participating in the AMR Register initiative. Shinogi as a company is committed to helping to fight antimicrobial resistance, both by developing new antibiotics, but also by supporting good antimicrobial stewardship. As part of that commitment, Shinogi is a signatory of all the major global declarations on AMR. We also continuously monitor the activity of our antibiotics via multinational resistance surveillance programs and are keen to share that data, supporting wider public access to both our surveillance and clinical trial data through initiatives such as the AMR Register. Shinogi has supported 2 large multinational surveillance programs. The first, Sidero-WT, ran for 5 years from 2014 to 2019 and collected a total of 45,785 gram-negative bacterial isolates from a range of clinical sources and tested them against a panel of 12 antibiotics. We further characterised carbapenem-resistant isolates to look for molecular mechanisms of resistance. The Sidero-WT data will be made publicly available through the AMR Register. Since 2020, we've also participated in the well-established Sentry program run by JMI Laboratories, which provides similar data to Sidero-WT and can also be publicly accessed through the Sentry MVP portal. Finally, a quick snapshot of the kind of data that's included in Sidero-WT. It contains microbiology data on nearly 50,000 clinical isolates collected from 100 laboratories in 13 countries in the Northern Hemisphere. The isolates include over 30,000 Enterobacterales, as well as common non-fermenters such as Pseudomonas and Acinetobacter. Isolates contained many multi-drug resistant isolates, including 13% which were resistant to carbapenems, and included a range of different carbapenemase enzymes. So on behalf of Shinogi, I'd like to wish Vivli and the AMR Register the best of luck and great success for the future. Thank you.
Venatorx Pharmaceuticals is committed to developing novel antibiotics to treat patients with multi-drug resistant bacterial infections. Our global surveillance dataset draws from over 300 hospitals in over 50 countries and includes more than 18,000 isolates, among them carbapenem-resistant Enterobacterales and NGR Pseudomonas aeruginosa that cause difficult to treat clinical infections. Our data, which are of industry standard quality, are based on pathogens that are collected consecutively using a prevalence design to sample the current distribution of AMR phenotypes by species. This helps identify less common and emerging AMR phenotypes due to the global reach and size of the dataset. We want to take this opportunity to thank our partner, IHMA of Schaumburg, Illinois, in the US, for their collaboration in executing our global surveillance program. Venatorx believes that providing access to these data, together with comparable data sets from other contributors, will support research to address the challenges of AMR. Since the activity of standard of care agents is assessed within these data sets, researchers can evaluate cross-resistance as well as multi-drug resistant phenotypes. As data continue to be added year over year, they may help further identify trends in resistance over time and in different geographies. Understanding AMR prevalence in different regions may also help identify those countries in which future clinical trials should be prioritized. Venatorx is proud to partner with Vivli and contributed to the AMR Register to enhance and centralize access to AMR surveillance across multiple data sets. This register is vital to support educational and scientific communications, as well as public health policies and initiatives around the globe.
I'm Dr. Nick Kartsonis, Senior Vice President, Vaccines and Infectious Diseases and Global Clinical Development at Merck Research Labs. Merck is thrilled to be a contributor to the AMR Register and is grateful to Vivli for leading this initiative, which will make important surveillance data more accessible to partners in the collective fight against AMR around the world. To address AMR, we must first understand it. AMR surveillance studies can help set a baseline in a country, region, or continent of pre-existing antimicrobial susceptibility patterns. Over time, these studies can be used to identify trends and spot early indicators of resistance. As a result, diagnostic tools can be developed, the responsible use of antimicrobials can be supported, and patient outcomes and overall population health can be improved. For more than 2 decades, Merck has been conducting the ongoing surveillance study known as the Study for Monitoring Antimicrobial Resistant Trends, also known as SMART. SMART is one of the world's largest and longest running AMR surveillance studies and has collected approximately 500,000 samples from over 200 sites and over 60 countries. It collects samples from the urine, from the abdomen, and from the respiratory tract, from participants with urinary tract infections, intra-abdominal infections, and pneumonia, respectively. The launch of the AMR Register will help Merck make the availability of the SMART program data more widely known, and in doing so, enabling researchers to use it to make a faster, smarter, data-driven decisions to help address the urgent threat of AMR. Thank you for your partnership and collaboration. We look forward to tackling this multifaceted challenge together.
Gemma, over to you.
Thank you, Rebecca. Good morning, good afternoon, everyone. It's really exciting to be here and to be part of the launch of the Vivli AMR Register. So thank you so much for inviting us. And I just wanted to take a few moments really to run through Pfizer's commitment to AMR surveillance and specifically our contribution to the AMR Register. For Pfizer, our long-standing commitment to antimicrobial surveillance is demonstrated through the Antimicrobial Testing, Leadership and Surveillance Programme, or ATLAS as it's known. And since the inception of ATLAS in 2004, we've been committed to the equitable and transparent access of our AMR surveillance data. And just to go back a little bit in time, Pfizer was a founding member of the first iteration of the AMR Register when we made our surveillance data available on the site that was developed as part of the Wellcome and Open Data Institute collaboration, launched a number of years ago, the predecessor to this AMR Register launched by Vivli. And it was this commitment that was really recognised by the Access to Medicines Foundation. And we're really happy to have witnessed the evolution of that project and the development of this new platform on the Vivli site launching today. And we're also really happy to see that other companies are engaging on this issue and making their data available so that the power of industry surveillance data can be combined and realised. I cannot emphasise enough how critical the transparent sharing, use and reuse of surveillance data is to advance our efforts in research and public health responses to AMR and to ultimately help protect patients worldwide from drug-resistant infections. It's data that will underpin and drive action at global, national and local levels. Next slide, please. So ATLAS is the largest available data set of AMR surveillance data, and it represents 18 years worth of data from over 70 countries and over 800 sites, providing data for more than 700,000 isolates. And the research and public health community can gain access to this data to monitor changes in resistance for particular drug-bug combinations and detect changes over time. Next slide, please. In a pioneering move more than 2 years ago, Pfizer became the first company to add their antifungal surveillance data to the site, and we have more than 12 years worth of antifungal data from over 42 countries and 145 sites, and all of this data will be available on the AMR Register. So we're really proud to be part of this collective effort to bring surveillance data from industry together, to make it available to all. And thank you to Vivli for this opportunity, and also to Wellcome for supporting this initiative. And we really look forward to seeing the results of this effort translate from the research and health provider community to the patient. Thank you.
Thanks so much, Gemma. Turn it over to Carla.
Great. Thanks so much, Rebecca. And I just wanted to build on what Gemma was saying, but in my role, I have a little bit of a different perspective than some folks today. I'm the Bioethics and Strategic Projects Leader, and I'm responsible for clinical trial data sharing at J&J. So I come at this from a very passionate perspective about sharing data, and really appreciated Tim's comments at the opening, and want to build on what Gemma was saying of that real landmark effort by Pfizer to share ATLAS has really created a lot of momentum around this. And Rebecca, congratulations to you and your team for getting this across the finish line. I know it required a lot of flexibility and coordination among a lot of different stakeholders, and the realization of the register is truly a terrific accomplishment. So if you can go to the next slide, I'll be speaking a little bit from the commitments from a clinical trial data sharing perspective and hitting some highlights of the data set that we are very proud to contribute from Johnson & Johnson, and then share a little bit about that. So we have a long history of a commitment. So this was really seen as an important project and an important effort to really advance public health, and just couldn't agree more with the comments from colleagues in their videos and presentations about how this is an important piece of bringing together the data into a central place. And I've already seen some of the questions in the chat asking about the ability to combine and bring together in a common place. So I think that's a real differentiator in what this register has been able to do. We share clinical trial data from our interventional clinical trials through the YODA project and cross-list them on Vivli. So have a strong appreciation for the value that this platform can bring to bring together a lot of different stakeholders, industry, academic, and different organizations to be able to share these types of data. The data that we are actually sharing is from an antimicrobial resistance surveillance study for our multi-drug resistant tuberculosis product, Certuro, also known as bedaquiline. It was the first drug with a novel mechanism of action to be approved by the FDA in more than 40 years, when it was approved back in 2012. And in 2017, we implemented this drug resistance emergence assessment in MDR-TB called DREAM. And this is the data set that we are really thrilled to contribute. And if you see there, it was by mid-2018, we'd have thousands of bacteria recovered from TB patients from 10 countries at the time. And if you flip to the next slide, you'll actually see that we ended up with isolates that were gathered from 11 countries. And the primary objective of the study was to determine the level of susceptibility of the isolates to bedaquiline. And you can see the results there in the publication at the bottom. And so we are perhaps offering a humble amount of data sets and isolates, but are looking across our programs to really see how we can continue to support this effort, and are incredibly proud and privileged to be included in this initial group. And just thank you again to Rebecca, and congratulations to all of you for this.
Thank you so much, Carla, for your support and contribution.
Hello. Hi, Rebecca.
Hi there.
I'm not going to start. This is Karen from GSK. I'm not going to start the video because of the the issues we'd had earlier with bandwidth. Apologies. I'm in India at the moment, and I don't have a particularly stable connection. Just wanted to say that really sort of like, we feel that sort of being part of this AMR register is really, really critical. As we all know about the threat of antimicrobial resistance. At GSK, this is an area that we're really, really passionate about. And we're proud to have been consistently being recognized as an industry leader in the AMR benchmark. We're working to combat AMR in many ways, including through our research and development, supporting both the prudent use of existing antibiotics and working with partners and working in the pipeline. And really by harnessing all of these, including the open sharing of data, we can prevent and mitigate AMR and work to get ahead of AMR together. So the data set that we're sharing is the SOAR data set. And SOAR, the Survey of Antimicrobial Resistance, it works in, it's in the area of community-acquired respiratory tract infections. SOAR itself has been going since 2002, so it'll be 20 years this year that this study will have been ongoing. And before that, GSK supported something called the Alexander Project, which started in 1996, I think it was, so even before that, so been going a long time. But really sort of like we focus now on sort of like Streptococcus pneumoniae and Haemophilus influenzae, looking at susceptibility to a panel of about 17 antibiotics. And we really feel this is critical to share these data. Next slide, please. So at the moment, we've shared our one data set, which is sort of like the data across sort of like 9 countries, but we've collected data across the Middle East, Africa, Latin America, Asia Pacific, the Commonwealth of Independent States, and Europe. And we'll be sharing further data, including from the current study that we have running in another sort of 12 countries, plus another 16 country data sets from a recently completed study. And really we're committing as GSK to share all our data going forward from SOAR and the next generation of SOAR, plus we hope to share all the data from the sort of the research that's being done on our pipeline of antibiotics. So thank you. And thank you to Vivli for driving this, and we're very, very proud to be part of it.
Thank you so much, Karen and everyone else that is participating.